Okay. Thank you all very much. It's actually been an honor to be here. I'm new to town. Essentially, I started a USDA position here in the summer of last year. So I'm a new plant physiologist with the Water Management Group. I won't be talking about any of the work that I've started. This is my first field season here. I'd be more than happy to talk to people about what I'm currently doing. I'll be showing you some stories from some of my past work. Some of it is still current. Things that I'm working on. One of the stories is actually a paper that should be out any day now. But I'll be more than happy to talk maybe at the break to people about some of the deficit irrigation studies that we're working on with corn and sunflower. So root traits, there's quite a lot of interest these days on root traits, and specifically in breeding for root traits. People have been talking about breeding for root traits as kind of like a new frontier. And clearly, roots are very critical. Root functions are very critical to plants. But really, when it comes down to it, functional traits are still very poorly understood. Root shoot ratios, root traits, root dynamics, development, it's all very dynamic. There's a lot of plasticity there. But as we'll see in the course of my talk, there are also some patterns that are definitely clear. Roots represent a high carbon cost to the plant. So in thinking about whole plant strategies, crops and yields, but both the costs and the benefits of what's going on below ground is certainly of a lot of interest. So in my talk today, there's two parts that I'll be presenting. One of the first part, we'll take a look at a story of root traits linked to plant strategies, whole plant strategies, and a story of root trait evolution. And the second part, I'll go into some work on one crop, grape, and talk about the effects of the plasticity of root dynamics in response to plant and environmental factors. So, okay, so let's cut off a little bit. Interesting. There's been a lot of interest in root traits. When I first started my dissertation research, I actually sat down and I had all these ideas about what I wanted to study and all these hypotheses that I wanted to explore. And it turned out, as I sat down with the literature and started trying to plan a thesis, there was almost no information available on root traits. So there were certainly a lot of ideas of suites of traits that should exist, but nobody had actually gone through and validated that or actually explored at all. There was no validation of suites of traits that were actually correlated with plant growth strategies. And there was actually more shocking than that. There was very little information about roots that you could even ask very basic questions of how broad is the range in root diameter among species. You know, when people look below ground, they would just look at this mess of roots and just say, okay, the root system, as if it was all one type of root in the root system. There was at the time, very little understanding of the complexity of the root system at all. So when we're trying to explore questions just of, you know, what traits are available, there's a complexity to the root system that we need to start understanding. And you see here that, and Amelia's talk yesterday, there's a complexity to the root system. All roots aren't just one. There's parts to the root system. It actually mirrors the above ground part of the plant, where you have these ephemeral tissues that are very, similar to leaves above ground, leaves versus stems above ground. You have also below ground. You have the fine root system, and you have the coarse root system, the structural component, and these fine roots that are doing most of the work in nutrient acquisition, resource acquisition. In woody plants, the fine roots are this ephemeral, non-woody portion. And it turns out we are a lot more specific these days, and when we talk about the fine root system, we're talking about the first and second orders of branching. And it turns out that the root diameters are a lot more consistent, and you get a jump in root diameter classes as you moved into the third order roots. And the third order roots, you start getting into roots that are going to become woody eventually. But they look different, even when they're not woody, they look different right from the beginning, from when they're first produced. They're coarser, anatomically they're also different. In herbaceous plants, you also get a very similar difference, as Amelia showed yesterday. You have, you'll also see a jump in diameter class between the fine and coarse roots. And they're also structurally very different, although it's a little bit harder to separate the physiological activity sometimes in herbaceous plants. So when I talk today, when I start showing you data about fine root traits, I'm referring here to this ephemeral portion of the root system in woody plants. And as you can see from this picture here, this is actually a woody root that we buried in a root bag and came back later and harvested. And you see this great proliferation of fine roots. If you look at plants and their root systems, it turns out both in woody plants and in herbaceous plants that there's a lot more root length in this fine root portion than there is in the coarse root portion. So the coarse root portion that's for structural support, it's this fine root portion that's doing the work. And that's where there's a lot of, it's a thinner roots, and there's a lot more of them, and they're doing the main work for the plant. Okay, so in the first questions that I tackled, a quick summary of this work. I started by trying to identify patterns and root traits associated with plant growth rate. So what I did was I sat down and I identified in the eastern forest where I could contrast fast and slow growing species within family. So I started working with seedlings in the greenhouse in 3 families where I could find an early fast growing pioneer species and an old growth late successional species in each of 3 families. And I started taking a look at some of the ideas that people had proposed at the time, root morphological traits, physiological traits that were associated with soil exploration and acquiring resources for fast growth. And that was that, well, of course, then tissue that maximizes surface to volume ratio, so thinner roots, that would have greater specific root length. You should see greater specific root length and finer roots in fast-growing species. And we did, in fact, see very clear patterns of that. So in the seedling work, in contrasting fast and slow, in 3 families, the fast-growing species is in the dark bars, and they had consistently morphological traits that complemented those ideas. Here I moved later on to harvest roots from the field. In the field I had 11 species actually in 5 families. And it was the same pattern. This is actually shown as a reaction norm with fast-growing species on this side and slow-growing species. This right here is specific root length, how much length you have per unit biomass, and you have more length per biomass you have fine. It turns out that this morphological trait is associated with the plants having thinner roots so that they can explore more soil. I also looked at physiological measures. It turned out this part of the story, I also modeled these traits to try to explore both costs and benefits of this tissue for phosphorus acquisition. And it turned out that in seedlings, the physiological responsiveness of the tissue was also, there was physiologically more active tissue in the fast-growing species, when I started looking at the mature tree traits, their fine roots, there was no difference in phosphorus uptake and respiration rates. And so it gave my explanation for this was basically that, that morphological traits, although there is plasticity in morphological traits, that morphological traits were more inherent, more hardwired into the plants, and the physiological traits depended more on the physiological state of the plants. So the seedlings is where you have these early successional and late successional species, their growth rates are most different at the seedling stage, and as you get into mature plants, their growth rates are more similar. So this seemed to be a very nice story, this pattern between fast and slow growing species. But I also wanted to address that question of what the range of trait variation there was in these root traits within the entire forest community. So I sampled 26 species in the forest and took a look at their root traits. And again, there was a very large range. It's almost a tenfold difference. There's a very wide range of variation. It's actually quite astounding when we were sampling. Of course, it's very intensive to measure so many traits in the field. So I had a whole crew of undergraduates that would go out with me and collect roots. And it's pretty amazing that these students that had no experience identifying plants above ground, in a couple of weeks, they could identify species by their root traits. They're that distinct. And there's a huge range of variation. Even within this northeastern forest, there's a huge range of variation. We have roots as fine as 1/10 of a millimeter to over a millimeter and a half diameter. So it turned out when I took a look, there's fast-growing species that have coarse roots. So this here now, the trait that we have mapped is specific root length. And here is the magnolia clade. Well, there's still some very fast-growing species in this clade, and yet they all have very coarse roots. So this idea of fast and slow-growing species having finer and coarser roots was a pattern that we only see within close relatives. And if we try to look more broadly across species and their growth rates, we see a very different pattern. And it turns out that there's, what we were seeing seemed to be a phylogenetic pattern. There's a consistency index of about 0.44. So what that's telling us is that about half of the variation that exists is actually mappable, is actually consistent with a phylogenetic tree. So there is still variation within some of these clades. That's the other half of the variation. And if we looked really closely at that, we would see things popping out like there are still coarse-rooted species in some of these clades. For example, black walnut here has close relatives with other species that are very fine-rooted, but black walnut has these other strategies for competing below ground. It has very coarse roots, but it has all these allelopathic strategies. So there's some other very interesting stories here, but overall it was quite interesting that there seemed to be this phylogenetic signal, and there was this phylogenetic pattern. So this forest species, when we looked in the northeastern forest, you're talking about basically looking at a community that has already undergone environmental filters. So later on, after doing a postdoc, I had an opportunity to write a grant with a colleague. And so we started in that grant, we actually were addressing questions of root trait plasticity, but I also went back to this question that I had of trying to look at these phylogenetic patterns of root traits and broad patterns. It seemed appropriate to address these questions with tropical angiosperms, because after all, that's where the epicenter of plant diversity it's considered to come from the tropics. So I sampled here, it's 20 species sampled from botanical collections. So where we could go, we actually used the New York Botanical Garden and a botanical collection at Columbia University and sampled 20 plants. Right here, we have the angiosperms. There, you'll see here, it's very hard to read. I'm sorry about that. But you'll notice, or I'll point out so that you can notice, that the angiosperm diversification mainly occurred in the Cretaceous period, and we also included some outgroups, some non-seed outgroups for contrast as well. So this now, actually it's not labeled, but this is root diameter as opposed to previously we were looking at specific root length. The darker lines are basically showing, and they get lighter across the phylogenetic tree. We have finer rooted species that diversified more recently, and the more basal lines had thicker roots. And it's not as distinct from here because the colors aren't showing up quite distinctly, but if we take that data and look at it a different way, this is actually, this 0 here is the root of that phylogenetic tree. And this is time in millions of years from the root of that tree. And this is it's an ancestral reconstruction at each node, reconstructed what at each one of those nodes and distances from the root of the tree, what the root diameter is estimated to be. And there's a very clear pattern with time that root diameters were getting a lot finer. So this is a paper that that is about to come out. We've already done the page proofs for. It should be out any day. Very interesting here, what's going on. Here are our basal non-seed plants with very, very coarse roots. And here is where we start seeing, this is basically that angiosperm diversification that's occurring during the Cretaceous period. So during that Cretaceous period, there's a few things going on here. So at the beginning of this Cretaceous period, these angiosperms would have been minor components of the environment. They would have been living in this dark and disturbed habitat, these very wet, dark and disturbed riparian areas, essentially. And it's pretty well documented that at the time, CO2 concentrations were starting to drop. And there's some very nice work. Taylor Field and Tim Brodrib have shown that corresponding to this drop in CO2 levels, that there's greater stomatal densities. The plants were evolving greater stomatal densities. They were, these angiosperms were evolving higher leaf venation densities. And it turns out from our work that we're also seeing that the plants were evolving thinner roots, potentially, so thinking about then why and what selection pressures would have led to this. These thinner roots would have potentially less cortex, potentially less impedance to water movement. If they were, these angiosperms would have been, they were moving from these wet habitats into upland habitats and competing, now starting to become dominant components of these early forests that were dominated by tree ferns and these drier habitats. In order to become competitive and dominant in those habitats, they would have needed to compete for resources and support fast growth with resources below ground. So they were starting from the wet habitats that they were coming from. They were starting to develop these finer root systems, potentially that allowed them to become more competitive for resources below ground, and also to support higher photosynthetic rates. As CO2 was dropping, they would have needed more water and nutrients to support higher photosynthetic rates with more stomates. Okay, so there's this other idea that that by producing thinner roots with longer specific root length, that they would also have allowed plants to produce more roots. They could invest in a bigger root system as far as root surface area is concerned and root length is concerned. And it turns out that it's very difficult to find information. Most people that look at traits, root length density is a very difficult trait to look at, and so most people don't. But there is data available. We regraphed some data that we found in Withington et al. From 11 species, which actually shows quite nicely that there certainly is suggestion that there's a trade-off, that when plants that have a greater specific root length, they also have greater root length density. So they have a larger root system, a denser root system. So as plants then would have evolved into these habitats, they potentially, with evolving finer roots with greater specific root length, longer specific root length, they may have been able to support a larger root system as well. Okay, so just to wrap up this section, some of my work with root traits suggesting that within close relatives there is, that we do find longer specific root length and thinner roots that allow potentially allow fast-growing species to explore soil pockets more quickly. But it seems that it's root morphology rather than physiology that is hardwired into the plants. And there's this evidence from this evolutionary story that that plants, angiosperms, early angiosperms, may have evolved thinner roots with longer specific root length to potentially increase water uptake and transport. Okay, so I'm going to switch gears a little bit now and talk about, now within one species, grape, Concord grape, some plastic responses in the dynamics of roots. And this project came about, Alan Laxo at Cornell had initiated a study about 6 years before we started looking at roots. He had initiated a study to look at above ground pruning and irrigation effects on yields, because farmers in the areas, this is the grape growing region of New York. It's juice grape growing region. So farmers are not getting the cost for their yields that wine grape producers get. They're very interested in minimizing their costs, and the farmers in the area were very interested. They historically had recommended a pretty severe pruning treatment, but farmers were going to almost this unpruned treatment completely. There's just a hedge cut at about 1 meter off the ground to keep the vines off the ground. There's concerns that we've done so much breeding to Concord vines that they can produce such a heavy load that we might need to start suggesting to farmers that they need to irrigate. Historically, this area didn't irrigate. The farmers would have experienced maybe 1 year in 5 where there was drought, but the decreases in their yields weren't enough to support their costs of putting in irrigation. So they essentially just weathered those. But as they started moving to these essentially non-irrigated systems, there was concern that that the vines would actually hurt themselves and not support root growth to sustain the yields, and that they would start having major problems with alternate bearing vines. And some farmers had started experiencing these alternate bearing vine problems. So this experiment was initiated, and then several years later, 6 years after the above ground treatments were initiated, Alan was interested in having collaborators look below ground so that he could get a whole plant view of what was occurring. So there's a couple of different theories that, that we were addressing with this work, and one is this balanced growth theory, this idea that plants put resources to offset their most limiting resources. So we can flip that around and basically ask, if we then irrigate, do we compensate for the amount that the plant would otherwise need to put? Would we see a smaller root system in an irrigated vine if the irrigation compensated for some of those limiting resources? And then this idea that there was this very strong idea in a lot of the tree fruit literature that there's this competing sink, and that there's this competing sink between the fruit and the roots specifically, so that if you have a heavy yield, that the plant is not going to be able to put as much as it needs below ground. Part of that idea came from some studies. There were just very few studies of timing of root growth in several crops. This is in grape. The blue line here is the line for root growth, and historically, the only information that was known actually about the timing of root growth in grape actually just came from one study in South Africa, and that happens to be a Mediterranean climate. But they had observed that there was a bimodal distribution in root growth, that there was an early spring flush, and then there was a fall flush, and there were no roots produced in the summer. And the idea was that, well, that's because the vines are ripening fruit, and that there's competition, so that there's no resources left for root growth. So here we were in New York, where it's not a Mediterranean climate. You actually get a lot of rain often in the summer, in most years, although we had 2 dry years. We initiated this experiment, was essentially a block design. It was a 2x2 factorial with 2 levels of pruning and 2 levels of irrigation. The axis actually, so our experimental unit was a set of 5 vines with a buffer vine on either side, and Minirhizotron tubes went in the ground in between those center vines so that we could take a look at the roots. The study lasted 4 years, and it so happened we very conveniently had 2 wet years and 2 dry years. We started with the wet year. We had 2 dry years in a row of increasing drought very nicely, and then a 4th year that was wet. So we took a look at the root growth with Minirhizotrons. These are clear tubes that go into the ground. We put 6-foot tubes in the ground at a 30-degree angle from vertical. And then you send a camera down. This is the Minirhizotron tube sticking up out of the ground. The camera goes down, or I guess this is the Minirhizotron tube. I can't see my own pictures. But the camera goes down the tube, and you record images along a column of windows, and then you sort the images at the end of the season so that you have a portfolio of windows that you can observe root growth in, those very small windows, about 1 centimeter by 1.5 centimeters. And you can watch the roots come and go. So it turned out that when we went to look for the effects of our treatments, we ended up having those 2 very dry years, and here we see dry year patterns versus wet year patterns. In wet years, there was no difference between irrigated and non-irrigated vines, and in the dry years, there was an effect, and it was exactly opposite of what we would have thought. We would have thought that with irrigation, that the non-irrigated vines would have actually had a bigger root system because they would have needed to forage for themselves for more water. And it turned out that they actually had a smaller root system, which was a bit of a surprise. If we look spatially how that root system was distributed, this is actually, again, our blue lines are the irrigated, and the red lines are the non-irrigated, and we have our 2 wet years. Our first year and our last year were the 2 wet years, and there's no difference in irrigation down the soil profile. This is down to a little over 1 meter. There's no differences down the profile. In the 2 dry years, it turned out that the dry treatment actually had more roots produced in the very shallow zone, and then as you went down the soil profile, there was in the first year there was a limited root production, 1 zone down from that top layer, which was about the 20 to 40 centimeter depth, and then in the second year, which was more severe stress than the first year, down to 60 centimeters, between 20 and 60 centimeters, there were almost no roots produced in the dry treatment. And yet, so the root production is here so that we can separate out the effects of our overall root system size. This is actually taken as a % of total roots produced. And what we're seeing then is that proportionally more roots were produced in the dry treatment in the shallow zone, and that's probably because there was an occasional rain event, and it probably only could wet down to 20 centimeters, and so that's the zone where roots were produced. So just to give a quick summary, basically that there's these ideas of root to shoot ratios shifting, but at some point we were working here in a very dry system, in a very sandy system, which dries down very quickly. And so there's a grain of salt that you need to take when thinking about how plants respond in the root to shoot ratio, that at some point when soil becomes too dry, it probably prohibits root growth. So although plants would perhaps could use a larger root system, could use more resources below ground, they're also not going to put those resources where either the soil is too dry or where there may be no water for them to take up. Okay, so then the timing of root production, this may be a little intimidating, but really I just want to show with this that if we look at the timing of root production in all 4 treatments, in all 4 years, you see very clearly that the main root production is occurring right smack in the middle of the growing season, where the hypothesis was that there would be, or where that other study had shown that in a Mediterranean system there was no root production. Here we're seeing all the root production. The main flush of roots is actually occurring right at the same time as the vines are producing their yields and ripening that fruit. Here that's that same data but shown as a rate, and here on this axis we have vine phenology and showing the root production as a rate per day, per window per day, because these stages of vine phenology span different times. But we see quite clearly that in the dry years there's very limited ability to produce roots in the dry treatments, but in the wet years and in the dry years with the irrigated treatments, they're producing their fastest rate of growth, of root growth, is occurring right there between fruit set and fruit development and fruit ripening. Okay, I think I'm going to skip this in the effort to wrap this up. So just in conclusion, there was no evidence of carbon limitation on root growth. And both the amount of and timing of root growth seem to be controlled by plant and environmental factors, and in this case it was canopy development. I actually neglected to tell you that the canopy development between these heavily pruned and minimally pruned vines was actually very similar. So peak canopy, peak light interception was the same between them. The one difference was that the minimally pruned vines actually produced that canopy a little bit earlier, and that's actually what was here in this slide, is that you saw then the roots also were produced almost a month earlier, and they were produced more shallow because the soils were probably warmer at that time. So I'm going to skip this, but this section of the talk was basically here just to remind me to tell you that roots, root morphology, there's an added complexity with root aging effects, and that there's been some work on that in terms of looking at age effects and aging effects on nutrient uptake. But there's a very, very interesting story with root strategies, and you can take a look at some of the work out of the Eisenstadt lab, looking at what they've coined it apples and oranges. There seem to be two different strategies for producing roots and their aging effects. And in apples, essentially you have, and grape is similar to apple, you have these fine roots that have a very short lifespan, and when you have median lifespans in apples about 30 days, and in grape is about 45 days. And then you have other species like oranges, and you have sugar maple as well, where those median lifespans can last more than a year. So these species seem to, apples and grapes seem to downregulate, or seem to lose root function, and the tissue dies, whereas oranges, citrus produces these really tough roots that seem to be able to downregulate function, and then and they can be in bone dry soil. Citrus is amazing. It can be in bone dry sand for months, and then as soon as they're re-wet, within a few minutes, regain function, upregulate respiration rates, and start being able to take phosphorus up again. So just to conclude, there are some broad generalizations that might hold between fast and slow-growing species, drought-adapted species. But there's a lot of complexity there when thinking about root traits, root functioning, and how that interacts with plant life strategy and resource acquisition. So we have certainly a lot more to learn before we're probably ready to start telling breeders what to breed for. But there certainly does seem to be at least the beginning of a story that we can start recommending some traits with, but a lot more work that needs to be done. And just then a caveat just to say that in any type of breeding for drought especially, you really need to know your system too. So with that, I'll end. I certainly had a lot of work doing all the work that I presented. We had funding both from the USDA, from NSF, and the phylogenetic analyses, and it was also funded by NESCENT.